Good morning, everybody. I'm very happy to be here. This is a fun opportunity for me. It's not that often that physician scientists like myself get a chance to talk about some of the things that I'm doing that may have an impact decades from now to an audience of people that are so affected and interested in what's going on. In the scientific community, these talks tend to get very technical, but I'm explicitly not going to do that and make sure that it's on a level that everybody can understand. If you don't agree with me, you could blame my wife. I gave her the practice talk a few days ago, and she said, "Oh my God, that was amazing. I understood everything." So if she's wrong, it's not my fault. I may use the pointer occasionally, and I'm not really sure it's work. Oops, that's definitely not what I wanted to do. Hold on. Yeah, if I do, it will be on this side, on my right. So I apologize on that side, but it's closer, it's easier to be direct. And just by way of further explanation, so one of the things that I do at Baylor is I take care of patients, and I have some patients here today, which is really fun for me and a privilege to see you. But I also run a big research lab, and that's why I was asked to give this talk. And I have several PhD students and PhD level employees in the lab who conduct research under my supervision. And the main goal of my lab is really to try to understand some of the earliest events that occur in glaucoma. I'm not really that focused on regenerating the optic nerve or bringing sight back. I have plenty of colleagues in the country who can do those things and do them better than I could. And I focus on the early steps. Can we find glaucoma? Can we stop it from ever being a problem? Those are the things that interest me. So with that in mind, I'll just remind you with this one question: what is glaucoma? Right? It seems such a simple thing. We focus so much on the treatment and day-to-day operations of people with glaucoma that we sometimes forget to stop and say, what is it? And really all it is a progressive optic neuropathy. But what does that really mean, right? We know what progressive means: it gets worse over time, and optic neuropathy means it affects the optic nerve. But really, in a practical sense, there's kind of 3 stages of glaucoma. And these are not exactly the same as the staging that we use for coding. This is more like a practical staging. There's an early stage of glaucoma where I can't tell if you have glaucoma or not. But I know that one day you might get glaucoma, or some people who will get glaucoma eventually, you're in that group. But I can't tell. The tests we have don't allow me the sensitivity to see it. And second, we have sort of that middle part of glaucoma, where many of the patients that we're treating fall, which are people who have optic nerve changes or cupping, like you may know about, and vision changes associated with that, everything from visual field changes to contrast sensitivity abnormalities to whatever it may be, an army of problems. And the third is the later stages, where you have really severe glaucoma. The optic nerve essentially is not working. The vision is very badly affected. And we're really going to think about this anatomically so you can get a sense of what it looks like. So I'm sure many of you or all of you have seen pictures of a normal optic nerve and an optic nerve with glaucoma. And if we look at it, we can see that a normal optic nerve has a tiny little cup. But in glaucoma in the later stages, we get this increased cupping of the optic nerve. We lose the nerve tissue. And then in a cartoon over here, this is the optic nerve here in red. And these are the cells whose axons or extensions become the optic nerve, and those are called retinal ganglion cells, or RGCs. And I'll talk about those quite a bit, but you see here in a normal state, it's all perfectly intact, but in severe or later stage glaucoma, you have fewer cells. They can't project through the optic nerve as well, and there's less information getting from the eyeball to the brain. So optic nerve really consists of specifically this part of one kind of cell in the retina. Retinal ganglion cells, or RGCs. And those are the cells that people are trying to repopulate when they're restoring vision in advanced glaucoma. And this is the cell that communicates all the information from the retina and sends it to the brain. So really, this is the cell we need to understand. What's going on? Okay, so with that in mind, I remind you of my premise. And then we say, in this early stage where things are undetectable and people see just fine, what in the world is going on, right? So the hypothesis that we took to start to ask this question was that risk factors for glaucoma might impact the retina at early stages prior to our ability to detect them. And because of that, we need better ways to find and even just describe what's going on. So what are the risk factors? And now we're switching over from glaucoma in general to specifically primary open-angle glaucoma. And the reason why I focus on this in my lab is not because I'm not interested in congenital glaucoma, or I don't care about genetics of glaucoma, or JOAG. All that is very important, and we know a lot about them. But we know surprisingly little about POAG and why it happens in the first place. We have all these correlations, but we just don't have enough to say, you're the one who's going to get it, or you're the one who's going to get it, and you're not. And I need information. So the risk factors that we have clearly established for POAG include eye pressure, age, and everything else, right? So really, age and eye pressure are driving what's going on. And we could focus on everything else as much as we want and find all these cool little things that are interesting and maybe helpful. But really, if we could understand why age and eye pressure interact to cause glaucoma, we could save a lot of people future blindness. All right, so how do I do this? Okay, so I do my work in animal models, particularly in mice, and we use a system, which I developed myself when I first started my lab about 15 years ago, to raise the eye pressure in the animal. And if you think about it, what do I want that pressure to be? So we know that in a person, the average pressure is 10 to 21, but we know that there are some problems with even thinking about a normal eye pressure, because we know people with glaucoma sometimes have a really low pressure, sometimes really high pressure. It really can go anywhere. And a mouse has an eye pressure of about 10, just so you know. And we said, well, if I made that eye pressure go from 10 to 50, obviously that would be really bad glaucoma, but I don't think I learned a lot about the early stages of disease. So instead, I tried to make a model where the eye pressure would come up, but the retinal ganglion cells, or those RGCs whose axons go to the brain, weren't dying. They were alive, but maybe not totally healthy, and I could see what was going on before they started to die. And then the other way to do it is to use not just convenient adolescent animals, but older animals that we sit and wait, in some cases for even 2 years before doing our experiment. So these are time-consuming, somewhat tedious, and the work I'm going to show is all published. The first part of it is the first paper I published in my lab in 2013. I'll show a little bit about how we made that model, and then I'll take you through after that just some more recent information that talks about some of these interesting risk factors and how they interact in 2 papers that my lab has published, one 3 years ago and one about a week ago. So hot off the press, all right? So how do we make the eye pressure go up? So we had several lectures this morning telling us a little bit about the physiology of eye pressure, where the aqueous is produced, where it goes out of the eye, and how that drainage system, turning on the faucet, clearing the drain, all of that is related to the eye pressure of the eye. So what I decided to do, and again, there are other labs that do something similar, but this is my particular approach on it, was to block the fluid from leaving the eye. So the eye would make its fluid normally, but I would partially block the ability of that fluid to get out. And the way we chose to do it, we're to take these tiny little beads. They're like little spheres. They're a micron or 6 micron in diameter. So a micron is 1/1000th of a millimeter, right? So really, really, really tiny stuff. Mix them together. I put them in a glass pipette, which is a millimeter thick. So the inside is just under a millimeter, so tiny little spaces. And then with these beads in the front, which you can see in blue in the schematic, it's then followed by a viscoelastic, which is that clear gel we use during cataract surgery to hold things open so we could keep space. And that's mostly to help those beads stay in the eye. And then this is a schematic of what I did. So on the left, we have a top-down view of that eye, and on the right, we have a side view. So I make a small incision in the cornea of the animal using a tiny little needle, usually a 32-gauge needle, which is smaller than what we use on patients. And then I slide this glass pipette tip into that hole which I created. I inject the beads, I inject the viscoelastic, and it pushes the fluid into the corners all the way around here, which is where the trabecular meshwork is located. And by putting it there near the trabecular meshwork, I could stop some of the aqueous from leaving the eye. And accordingly, we end up with this. And this is the first figure of the first paper my lab ever published, all right? And I'm still showing it in talks 12 years later. But here we have animals that did not get an injection of anything, and their eye pressure is right about 10. And we're tracking them here for 3 months. These are post-operative day, 0 through 85, so 3 months. And with that single injection, the eyes that got the beads went up to about 14 or 15, stayed there the whole time. So I have a 3-month window where I can do any experiment I want to do. And the reason why this worked out so well was when we started to look more closely at the retinal ganglion cells of these eyes, I found that they were almost all there, and that the effect of the eye pressure was not to kill the ganglion cells, but to injure them. And if you see over here, I could see at 6 weeks, we only lost about 7% of all the retinal ganglion cells. That means 93% were still perfectly there and happy. And at 2 weeks, the numbers were totally normal. So I now have a system where I could test anything I want after about 2 weeks of raising the eye pressure and try to understand what happens in these cells before they die. And that's what I've been doing for the last 15 years. Okay, so now let's talk about some cool new things that we found. So I remind you of my hypothesis that the risk factors for glaucoma will impact the retina at the very earliest stages before we can detect glaucoma. So the question is then, what should we look at? There's so many cells in the retina, there's so many different kinds of neurons, there's non-neurons, there's all these things. What do I focus on? So the only way to do that properly was to think about what we knew. So what was in the literature? What were people working on at the time where I was starting to think about this? And I'll briefly describe that there are some papers that had come out in the literature, some from my group and those associated with me, but also from others, including the much more famous than I, Andrew Huberman of Huberman Lab fame, focusing on 2 different populations of cells in the retina that seem to be incredibly sensitive to eye pressure. The one on the right here is a specific kind of retinal ganglion cells. These are retinal ganglion cells that, for whatever reason, and that's beyond this conversation, they don't respond to light. They respond to when light goes away. So they're not doing anything when the lights are on, and then when the lights turn off, they start to integrate information. The other type of cell that was affected were called amacrine cells. And amacrine cells are what are called interneurons. So these are neurons in the retina that help modulate what's going on and make sure that the clarity of the information getting to the optic nerve is as good as possible. So with that in mind, what did I know about these cell populations? So turns out that if I look at a retina here, and these are the nuclei of a cross-section of a retina from an animal, it has multiple layers. And at the top layer over here is where all the retinal ganglion cells reside. And at the very front of this second layer here, called the internuclear layer, that's where all the amacrine cells reside. And it doesn't take a lot of understanding to realize that these are pretty close to each other, right? They're not in the opposite ends of the retina, they're right near each other. So maybe there's something in that local area that's important. So I'll show you here a schematic I made that puts together the different neurons in these areas. And the most important ones for us to focus on are these blue ones here, which are the RGCs, and these green ones here, which are the amacrine cells. And it turns out that the off RGCs and the RGCs that respond to light, which are called on RGCs, are structurally different. And the off RGCs have these longer extensions called dendrites, which communicate with amacrine cells right here in this little area. So they're in the same place. So what else is there, was the next question I asked. And it turns out there's a blood vessel and a set of capillaries that runs right through that area of the retina. And the retina, as it turns out, has 3 capillary webs that get all the blood supply to the cells. There's a cell population that's right near the retinal ganglion cells, called the superficial retinal capillary plexus, or SRCP. There's this other plexus that is sitting right in the area that we're most interested in. It's called the intermediate retinal capillary plexus, or IRCP. And then there's one that's kind of hanging out here over here, closer to the photoreceptors. It's called the deep retinal capillary plexus. So that's a lot of information to take in. So I want to pause for a second before we go any further. But one might think that if glaucoma is a disease of just retinal ganglion cells, that this blood vessel at the surface might be the one that's most important. But it was not lining up with the other data. So we started to think, well, how can we tell these vessels apart? How can we understand what they do? So I went into the science, and I found out that there was really not a good way to do this. So without a good way to do it, how could I test that the IRCP was the link? So the answer was to make a way to do it. So I made a whole system in my lab to understand and assess the capillary structure of the entire retina of an animal with or without glaucoma. And I'm going to show you that in a minute. The way we set up the experiment, which is going to be the rest of this talk, is by taking animals that were young, about 12 weeks old, which is the same as about 20 or 18 years old in a human, and old animals that were 18 months old, which is about the same as being 60 years old. Okay? So adolescent versus old. I'm not 60 yet, so I can still say that. Okay? The goal was to take both of these sets of animals, increase their eye pressure in just one eye, which is also called ocular hypertension. You'll see OHT written sometimes. That just means elevated eye pressure experimentally. And compare the old or the young animals with their normal condition and their eye pressure condition. And then I could see the effect of eye pressure on a young animal and how it might differ in an old animal. And I could even just process how aging affects the retina in general, something we don't really know that well. And then made a system to quantify the blood vessels and to image them. So when you quantify blood vessels, you get this really cool pattern, which is just like an OCTA that you may have seen, which is a OCT angiography, which is a human test to look at all the blood vessels. But we did this using post-mortem antibody staining and testing and things like that. So this is done after the animal has finished the experiment. But I want to just remind you that a blood vessel is not just a tube. It's a tube that's surrounded by something called a basement membrane. And the basement membrane consists of proteins and sort of sticky things that hold things in place and allow for movement of fluids in and out of the blood vessel in a controlled way. And that's important because anatomically, it turns out to be one of the areas that's most affected by eye pressure. So here's an example of what we can do with this approach. So this is a figure out of the recent paper. And what it does is it shows that whole map of all the capillaries of the retina in a young mouse with glaucoma, a young mouse with glaucoma, an old mouse, and an old mouse with glaucoma. And it doesn't take a lot to look at the difference here. And you could tell all this is at the same exposure, the same magnification, everything's the same. But these two look different from these two, right? There's not as much blood vessels. And we could quantify this. And indeed, glaucoma itself in a young animal does reduce the overall capillary structure of the retina, and then there's a big drop with aging, and then not really much of an additional effect of eye pressure once the aging has kicked in. So that was really interesting, right? We already see a big effect that nobody had ever found in how blood supply to the retina is affected by age and by eye pressure. But this is not enough, because it doesn't let me look at the three plexi that I care about. I can't tell the SRCP, the IRCP, and the DRCP from that low magnification image. So it turns out that if I zoom in at very high magnification, we could quantify not just how many blood vessels are there, but everything about them. How thick are they? How much area do they take over the retina? How many brand points do they have? And this is exactly what we've done to understand a little bit better what's going on. And this schematic just takes you through how we do that. So we use two different software programs to take our image, basically play around with it, and then end up with a way that we could quantify everything that's going on. Okay, so what do we find? So in our young animals with glaucoma, we found in a surprising moment in my career where my hypothesis was actually correct as opposed to incorrect, was that the IRCP had a big effect on the number of branches and junctions in the capillaries just in the young animal and just in that one area, exactly where we expected it to be. And with that, we looked at all the other parameters that we could quantify, and we saw the same thing. It's not normal in the IRCP, in the intermediate retinal capillary plexus, and totally fine everywhere else in the retina. And this is very exciting because it told us that we're onto something. We looked a little further, and now this is why that comment about the basement membrane is important. When a blood vessel gets injured or stressed, one thing that it can do is it's called remodel. It will go away and go in a different direction or a different area. And this happens during neovascular disease. If you have macular degeneration and you have to get injections, that's exactly what's going on. But it's also a normal process that happens under stress. And I can find that with my experiments, because what will happen is I can still see the basement membrane in yellow, but the blood vessel will be gone. And by using the right combination of antibody staining in a retina, I could see areas like this, where we have a little yellow thing, meaning the basement membrane is there, but the blood vessel is missing. See that? And there's a couple examples of that over here. And then we could count them. So medical students are great for that. So I've had a few medical students who joined the lab to count everything for me. And of course, in our young animals, what did we see? A huge increase in these abnormal acellular capillaries only in that one area, but not everywhere. So all consistent, okay? So then I went further and we looked at aging. And now I'm showing the data a little differently, so I'll just kind of walk you through it. This is now comparing an older animal to a young animal. So everything is benchmarked to the young. So this is like 100%, okay? So if you have an old animal, we could say, are the junctions in this particular plexus, are there more or are there less than what we'd expect from a young? If it's less, it'll be under 100. And if it's more, it'll be over 100. Pretty straightforward, right? So we could track everything, all the plexi and all the parameters. And what do we see? We see changes everywhere. Just aging itself causes the vessels to be a little shorter, a little less branchy, but thicker. So the vessels themselves are changing with age. And maybe that underlies some of the susceptibility of aging to glaucoma, right? A reasonable thing to consider. We looked at the same thing. We looked at eye pressure. And again, there's a lot on this slide, and it's not meant for you to see every little bit of it. But what I find is if we look at the superficial, intermediate, or deep plexus in young animals or old animals with glaucoma, and now we benchmark to their normal eye, we see the same kind of thing. There's a big reduction in general in all of the vascular complexity just caused by aging and eye pressure together. But if we look at each individual plexus, it's always the intermediate retinal capillary plexus that's most affected. So with this, we went to the last step, which was to do the acellular capillaries, which I showed you before, where the blood vessel moves away. And the same thing, very high numbers just with aging in the intermediate plexus, but not in other places. And they're all affected in glaucoma. So if you give glaucoma to an old animal, there's massive turnover of the blood vessels, and they start to pull out of their basement membranes. So just giving a mouse a 4 millimeter of mercury increase in eye pressure at a 12-week-old animal or a year-and-a-half-old animal has completely different effects on the vasculature, and they're much worse in these older animals that also show changes that nobody had ever seen just in the aged retina. Okay, so one last little bit I want to show you, which is our high magnification imaging of the retinal vessels. So another thing that we can do is we could collect some retinas, and we could cut them into slices and fix them with a fixative and take out all the fluid and all the stuff you have to do to prepare them, and do what's called electron microscopy, which is an old technique that's been around for over 100 years. We fire electrons onto the tissue, it bounces back, you record that, and you get this incredibly high magnification images. And you could see exactly this. You could see a blood vessel, the capillary of it, the basement membrane over here. You could see a nearby cell. You could check all the different layers of the retina and find the layer that we care about and look at the blood vessels directly. So by doing that, we found two major effects. The first was that basement membranes change as the animal gets older. And if you elevate the eye pressure on top of that, it changes even more. So this in the top left are the young animals in each of the 3 levels. And you can see this little strip of the basement membrane here, where the arrow is pointing. And then in an older animal, you can see, look how much thicker it is in every single space. If you compare this to this, or this to this, it's thickened everywhere. There's more debris in that basement membrane. And then the effect of eye pressure in this lever is to thicken it, but on these, it's to thin it. So the eye pressure can have different effects to either thicken further or re-thin the basement membrane based on injury. So the layer of the retina is all responding differently to the exact same problem. So there's a lot more complexity going on than I think we really realized in the past. And the last thing I want to show you is something called a tight junction. A tight junction is a protein structure that makes sure that a blood vessel doesn't just leak everywhere. It keeps it intact. And those blood vessels have this really nice little strip. You see it over here? This like double strip here of two lines running together. These are really normal, intact, tight junctions. But I'll just skip to the last one. Look at this one here, an older animal with glaucoma. This is the same area. They're all disrupted and abnormal. There's areas of loss so the blood vessels are much more leaky and much more injury prone in these older animals. So with all of this, I can conclude that eye pressure and aging have overlapping, but some distinct effects on the retina, that the vasculature is rapidly undergoing changes and remodeling, and that maybe stabilization of the vasculature is a way to treat glaucoma or prevent glaucoma from happening in the first place. So this is something that now my lab is starting to refocus on, ways to think about blood flow, about the vascular hypothesis of glaucoma, and thinking about neuroprotection in that context as a way to prevent future damage as opposed to prevent additional damage that's already occurring. And just want to acknowledge people in my lab. I've been very fortunate to have a lot of smart and happy people work with me over the years. In the top right, we see a much younger Dr. Frankfort hanging out with the lab 1.0. And this is our COVID shot. We still had to get together, so we did it in my backyard, and then some more recent iterations of the lab. And then, of course, none of this would be possible without money. So I always acknowledge my fortunate funding from the National Eye Institute, from several foundations, including the GRF and also the American Glaucoma Society. And then most importantly, particularly for this research, I always call it my grateful patients. I've had the fortune of having several patients, really almost a dozen now, be my partners in research and donate money to my lab to help me do these studies. And these aging studies that I showed were not supported by the NIH because I have not been able to get a grant for it yet, simply because it takes so long to generate the data, I didn't even have preliminary data to show them. So I used the donations from my patients to make these really interesting discoveries, which hopefully will support additional funding and maybe help us figure out ways to predict and treat people before they even realize they have glaucoma in the first place. That's all I have to share. Thank you so much for your attention.